Greetings and welcome to Houghton, Michigan, where I've been coaching the ski team at U.S. Nationals. We have reached that dreaded day where we depart tomorrow and we have to clean our vacation rental. So we are embarking in all of the interesting antiseptics and disinfectants within our vacation rental so that we can get this place clean. And you can see behind me that my athletes are working really hard at cleaning. They're all about it and really scrubbing away. Just kidding, but hopefully before we leave they will. So let's talk a little bit about antiseptics, disinfectants, the ideas of sterility versus sanitation, and that's actually where I want to begin, is by looking at the terminology sterilization. This means to remove all living cells, spores, and virolds. That is, we're killing absolutely everything. This is something that we cannot do with a disinfectant alone. So if you see somebody cleaning with, for example, this here Tilex daily shower, you can know that while they're sanitizing a surface, perhaps their shower, that they are not sterilizing that surface. Sterility is the removal of everything. The way that we accomplish that in the lab is to put it in the autoclave. We put it at extremely high temperature, 121 degrees Celsius, and high pressure. It's kind of like a huge pressure cooker with this incredibly heavy metal door that takes a little bit of your strength to just even get it closed. We raise that pressure cooker up to high temperatures, and that is how we get rid of all living things. Now, maybe I should put a caveat there. There are certain archaea that can live through this. They can take an autoclaving and keep on kicking. But for the most part, those things that we're worried about contaminating any of our glassware or media in the lab are completely killed by this high temperature and pressure. We, in fact, even do a spore test from time to time where we see whether or not spores, endospores, are killed by the process of autoclaving, just to make sure that our autoclave is performing up to snuff. So sterility is very different, and sterilization very different than sanitation. Sanitation means to lower levels of microorganisms to what is considered safe by some sort of standard. And if any of you have ever worked in the restaurant industry, you realize that there are those standards for the concentration of microbial life that can be found on any surface to enable you to continue to serve food in some establishment. So let's talk a little bit about the types of disinfection versus antiseptics. That is, maybe some of you have seen My Big Fat Greek Wedding, and in that show, it's really funny because the guy, one of the main characters, the father, he likes to use Windex on every kind of wound. So if somebody gets a cut, spray a little Windex on that. But really, that is the exact poor use, opposite use of what a disinfectant is for. So those things that are used to clean inanimate objects like Windex, those are disinfectants, whereas things that are used to clean your body surfaces, those are antiseptics. So disinfectants kill, inhibit growth, or remove microorganisms that are present on inanimate objects. A good example is the Tilex daily shower here that I found in our under our sink here at our vacation rental. My favorite of the ones that we have here is called borax, and the really cool thing about this is that it claims that it's so strong that it's a 20 mule team of cleaning. So you can see here borax. This again is something you only want to use on inanimate objects. I don't know about you, but I don't want a 20 mule team anywhere on my body. So this is a really good example of a disinfectant versus an antiseptic. Now, antiseptics are manufactured with living tissue in mind. For example, this Crest Complete antimicrobial toothpaste. This is something that you don't mind too much putting on your body. Another good example is Band-Aid Hurt-Free Antiseptic Wash. Here's something else I found under the cupboard in the bathroom. This is rubbing alcohol. Rubbing alcohol we commonly use on our body surfaces. So antiseptics manufactured with living tissue in mind. And remember, to help you remember this definition, sepsis means a large-scale infection of the body. So if we're using some substance to prevent that, then it is an antiseptic, stopping sepsis. So antiseptics, less harsh than disinfectants, and we don't want to use disinfectants on human or animal tissue. I heard this horrible story about a horse that got an infected hoof, and the owner kept dousing the hoof in Clorox bleach. And of course, this was doing more damage to the horse's foot than the actual infection was. So this is the reason we don't want to put Clorox all over our body, is because it's damaging the tissue perhaps as much as the infection is. So let's talk about those antiseptics and disinfectants that claim to be cidal. So you know the term cidal, suicidal, homicidal, right? It means to kill. So when we think about things that are bactericidal, fungicidal, algicidal, or virucidal, we can easily infer from that they kill either bacteria, fungus, algae, or viruses. Now, one of the most fantastic old-fashioned things that we have hanging around in the lab is something called sporicidin. And right off from that title, you get the idea that this kills spores. This must be some pretty BA stuff. That is to say, this is really, really harsh. In fact, it claims underneath to be a cold sterilizing solution. Now, this has historical connotation because Joseph Lister was the first person to ever perform a surgery using sterile instruments. That was a pretty good thing because it meant that there wasn't sepsis that came after the surgery. Now, this was unusual, and it was really a landmark discovery. No longer, though, do we use cold sterilizing solutions for surgical implements. Instead, they're autoclaved or something of that nature, and a lot of surfaces in an operating room are exposed to UV irradiation. But it's interesting to look back at this old school sporicidin. When you look down at the list of things that it claims to be cidal on, it's pretty whopping. So look at this. Besides being sporicidal, it's also said to be virucidal, tuberculocidal, bactericidal, fungicidal, pseudomonicidal. Man, this thing kills everything. And what's really funny is if you turn the bottle around, you'll see that it says kills 100% of all organisms. And then on the other side of the bottle it says, no need to wear gloves. It won't yellow your hands. So the irony, I think, is pretty inherent in that. I don't think I would want to utilize something that kills 100% of all organisms without having gloves on. But in any case, sporicidin was pulled off the market. It's no longer available, and there's a couple reasons for those. One of that is the claim to kill 100% of all organisms. So now on pretty much every disinfectant that you look at, it'll say something like kills 99.9% of all bacteria, so that they're not claiming to kill 100%, and it's true, they're not, right? They're sanitizing instead of sterilizing. Let's end today by looking at the mechanism by which these agents kill or inhibit the growth of particular organisms. And Joseph Lister's cold sterilizing solution, like the sporicidin, is a phenolic compound. Now, Lysol is also a phenolic compound, but it's highly diluted as compared to sporicidin. Phenolic compounds denature proteins and perturb cell membranes. That's the mechanism by which they kill microbial life. Detergents, why, these are another great example, and in fact probably my favorite example, because like dissolves like. So that is to say that if you have a detergent, it has a polar head group on it and a non-polar tail. Now, I'm going to draw that for you, and we'll pop out of this to another drawing program so that I can show you how that works. So here's a feeble drawing of a phospholipid bilayer, which we know comprises the kind of basal level structure of a membrane. And up above, I've drawn a couple of detergent molecules. Detergents, again, have this polar head group and then a long hydrocarbon tail. So notice how much they look like phospholipids. Phospholipids with their polar head group and their two long fatty acyl side chains have a very similar structure to a detergent, and therefore detergents dissolve membranes, right? Like dissolves like. And remember, that was one of the rules that we learned on the very first week of class. So detergents really emulsify membranes. They dissolve them and extract them. Halogens, these are things like iodine and chlorine, and these are used, we've talked about how they can be used in disinfecting water. Iodine, if any of you are backpackers, you might carry around iodine tablets with you. When we go on long runs with the skiers, we take some iodine tablets just in case we run out of water and we need to purify a little bit of water. So iodine does a really good job of oxidizing constituents of the cell. It also iodinates proteins, which wreaks havoc with their function. So reactions involving chlorine result in a lot of redox reaction, a lot of oxidative damage of cellular materials ranging from membranes to DNA, nucleotides. Betadine is an iodophore. Household bleach is sodium hypochlorite. So bleach is the one that we looked at here earlier. Wow, this bleach claims to be splashless too, but this is doing a whole lot of oxidative damage. Alcohols, such as isopropanol, remember looking at our rubbing alcohol, this one denatures proteins and extracts lipids as well. Hydrogen peroxide, an epic antiseptic, which we're now realizing does more damage to cellular tissues than we once thought. So it is not suggested so much as an antiseptic anymore, but it causes oxidative damage of cellular materials as well. You can see that with hydrogen peroxide. We've talked a lot about how that is an oxidizing agent. So with that being said, we're going to call this a wrap and make sure that you do review the virtual edge before getting started with your lab, and you go through the video talking about the procedure for testing disinfectants and antiseptics. Have a fantastic day.